Hello, it's V again for the Fundamentals in Statistics and Computation for Neuroscientists course at UCSD. And I'm just going to be giving a short little video about the remainder of the resampling methods I'd like to talk about. So last time we covered different hypothesis tests for distributions and a little bit about how you can use resampling to bootstrap your data or jackknife it to correct the bias in different estimates of descriptive statistics. We also went over a little example about how to use shuffling or randomization tests to calculate a more accurate t-distribution for your null hypothesis. So essentially, these were the questions that we started out with. We weren't sure how to model the data distribution, and talked a little bit about both these methods. We're going to continue talking about number 2 today. So I just wanted to continue the discussion about randomization tests because you didn't quite cover everything there is to cover here. So we talked about these first 2 questions, but this last question is actually quite a complicated one, and I'm only going to really talk about general themes rather than cover all the little nuances that are actually present for randomization tests here. So, what if you made up your own statistical measure? Well, this is actually just a general question that kind of glosses over all the things you could do. So we talked about this, making up the nullt distribution, and shuffling the labels. One thing that I forgot to talk about, which Burke pointed out in class today, is this little note that I have here, which is the precision of your p-value. So if you did your shuffling many, many times, you're going to eventually reach an asymptote with the number of iterations you get. So if you did 50 iterations, you're not going to get a very accurate null distribution. If you did 1,000 iterations, you would probably be pretty accurate having 30 samples each. But let's say you had 500 samples. You would need to continue shuffling for many, many more iterations. And depending on the precision of the distribution that you get, you will get different precisions of p-values. So if you only had 50 iterations, for example, you couldn't actually report a p-value of 0.05 because you don't have precision out to the hundredths place. So you just want to keep that in mind when you're reporting your p-values. You want to make sure that you have enough iterations that you're really getting the precision that you want. And you have to make sure to always double the number that you want to get the significant figures that you want. So if you wanted 0.05 to the hundredths place, you actually have to sample at least 200 times. So you would get both the hundredths place and then be able to round if you had something slightly on the edge. All right. Now, I just also wanted to point out that shuffling labels is essentially resampling without replacement. So before we talked about bootstrapping, which is sampling with replacement, this far right circle here. But when you're shuffling, you're essentially resampling without replacement. You're just, here N and K are the same values. So you're usually resampling by always getting the same number of elements out. You're just switching the order. So it's not quite right to call it unordered. It's randomly ordered in this case on purpose. You can really do this with any measurement. So if you'll remember, the resampling methods we reviewed both with replacement with bootstrapping and without replacement with permutation testing and randomization tests are all examples of Monte Carlo methods to simulate probabilistic events. And whatever measurement you choose to make, if you do it in an intelligent way, you can use resampling to help you derive statistics for your measurements. But importantly, unlike with bootstrapping, you cannot generalize the results of your randomization tests beyond the sample that you have. This is because you're sampling without replacement when you shuffle those group labels. And the p-value that you generate is only true for the distribution that you collected because you're still shuffling within that little distribution. And you cannot generalize to your population ever if you derive a result from resampling without replacement. So you really have to keep those two things in mind. It's usually more powerful to try to do a bootstrapping test if you can. So you can bootstrap your data and do a one sample test if your data allow that. However, if you are looking for an effect that requires a randomization test, you have to make sure to not draw inferences beyond what you can draw. And the only thing you can do in that case is to collect more data and try to replicate and then repeat your randomization test to see how it turns out. Okay, so these methods are really good for dealing with abnormal measurements, and I really want to give you a couple examples because they're actually quite common. We talk a lot in introductory statistics and hypothesis testing about normal distributions, and sometimes you can assume normality, but there are often limiting factors that don't allow you to assume normality. And even with more complex models, such as ANOVA, which can deal with multiple predictors, you can still sometimes run into problems when you're trying to analyze particular sets of data. So one particularly common thing that happens if you measure behavior, for example, or if you have an animal colony, is that you get some sort of change in a measurement over several temporal bins, which don't have an equal amount of time between them. So I actually just looked in the most recent issue of Nature and found this example because it's really so common. So in this paper, it's pretty cool. We're looking at the immune systems of laboratory mice and mice that were housed with other mice that had come from the pet store. So basically the argument was that their immune systems were different because of the environment they were exposed to was quite different. And here they're showing the level of these particular immune cells, CD44 cells, as a percentage of all CD8 T cells. And you can see that it's quite elevated in the mice that were exposed to the immune environment that was quite different from the lab mice environment. And for the lab mice, they stayed fairly stable throughout their whole time of measurement. What you'll notice here is that there's not the same number of time points for the green curve as there are for the blue curve. For example, we're missing this one here at day 83. And you'll notice that they sampled a lot more at the beginning and less towards the end. So you can imagine if you were going to try to fit an ANOVA to this, it would be kind of tricky and not quite right because you've got these levels to your factor which are not quite correct. They're not really equal levels. You also have a different number of points for each group. So it's not like you could do a ANOVA using group and time as your two factors. You could try to fit a GLM because you would be able to account for the differences in time here by just making sure your predictor was continuous and could handle that, except you see here that this relationship really isn't well predicted by a line. So fitting a straight line to this green curve wouldn't be correct either. Then some people would say, well, the differences are so large. Look at those error bars. Remember our discussion about error bars before? They don't always quite reflect what you think they reflect. And even if these are 95% confidence intervals, you can say that the percentages were different at day 107, for example, because the 95% confidence intervals absolutely do not overlap. And you can continue to do this as a repeated test across all the time points that you have. And then maybe correct for multiple comparisons. That would be okay to do. But you also notice here that all of these are percentages. So that makes it even trickier. Let's say the lab mice just had a different starting level of CD8 T cells. So is it really valid to compare two percentages when the denominator for these two groups is probably totally different. I don't know. It's really tricky when you start to think about it and you have all these qualifiers to measurements like this, especially with proportions and with temporal measurements. So one easy thing to always turn to is a randomization test. So as I said before, I actually didn't make this point yet either. The samples are also not independent. So especially with temporal measurements, measurement from day 8 is going to be correlated in some sense with the measurement at day 15, because it's not like you're going to flip a random coin on every single day and there's true independence from day to day. Generally, with anything that we're tracking over time, things are correlated because most things are correlated over time. As I said, this is also a bounded measurement, and what I didn't type out here was what I said before, which was that the proportion of CD8 cells relies on the denominator, which is the original number of CD8 T cells in both groups, which could differ across groups. And that's really tricky to account for when you're just looking at the single number, the proportion number. So what you could do is you could shuffle your time points between the two groups. So basically you'd be shuffling every single time at each day. So you could just shuffle, now this is the blue group, and then at the next iteration it becomes a green group for all 20 points here, or however many points they have in each curve. And that would give you a nullt distribution which keeps these temporal relationships intact. And then it would be more valid for you to do a paired t-test and then compare the value that you get for your t-statistic against the null distribution value as if there are no difference between groups. And as we talked about before in the previous lecture, the t-statistic actually doesn't depend on any assumptions of normality, it's just the distribution which does. So doing a paired t-test in this way would still be perfectly valid. You also could do something else in which you shuffle the time points within each group. So let's say just for the blue group, you shuffle between time points in day 107 and time points in day 22, for example. And that would help you to find any temporal trends that happen across this group. So in the blue group, you probably wouldn't find very much. This line looks fairly flat. But in the green group, you can tell that the data at day 8 look pretty different from the data at day 52. And then you could simply run a test which took into account this covariance. So actually I meant to write here an ANCOVA in which you would have these values as a continuous value. So you could take into account the fact that there were different numbers of days between each sample. So the thing about randomization tests and shuffling is that it's really powerful, but it really requires you to carefully consider your data and to consider how you want to draw your conclusion or what you're trying to test. And there's so many different ways you could then use the tools that you have to get to the statistical measure that you want. All right, here's another example of a different non-normal measurement that often pops up in the literature. So for a lot of different fields now, classification has become very popular. So this is just an example, a representative plot from a paper I will not name, in which images were shown to subjects and they tried to determine whether the image was before a mental rotation task or after a mental rotation task. So those were the two things that this classifier was trying to decode. And you can imagine, or you might have read many different studies which try to classify not just stimuli, but maybe state of the animal, try to see whether, for example, they're awake or asleep, which would be a really easy thing to classify, or whether they're currently receiving a reward or a punishment based on their neural data. There's all kinds of things that you can classify and use all the data from a bunch of cells to try to extract some sort of multivariate information from it. The problem is the classifier is very complicated, and it's not easy to derive a null hypothesis for your classifier. Because here you see that, there's two different groups. You would naively think that chance level was 50%. And if this were a perfect data set and the classifier was completely unbiased, then yes, we could expect that chance would be 50%. But that's typically not the case. So you also see here that actually these this y-axis is incredibly blown up, and all of these effects are maybe on the order of 2 entire percentage points. So that's 52% or maybe even 51% at the end of these error bars here versus 50%. If you read the figure legend for this little figure, they're plotting standard error of the mean, which means they're suggesting that these decoding accuracies are normally distributed, symmetric, and all of these other normal properties around this mean decoding accuracy, which they generate by doing their classification analysis many, many times, I believe in a cross-validated way. So, I'm sure you can spot many, many problems with this. When you cross-validate, for one, you're re-estimating the model on different subsets of your data. So it's definitely not independent. These samples are not generated independently. So it's not true that you could do a simple t-test versus 50% in this case, which is the first thing that I believe these authors report in this paper. And the other thing is that if you assume a standard error of the mean here, it's very easy to get some tight error bars because if your data doesn't have a ton of variance in it, especially across a huge sample of neurons, for example, the classifier will often end up doing similar things on every cross-validation fold. And just because there's not a lot of variance in the results of the decoder doesn't mean that it was accurately decoding. So you can imagine, for example, that it consistently gets 51%. But if it's fitting noise on every single iteration of the cross-validation procedure, and you're sampling a huge amount of data that has a very similar noise structure on every subset, you would still decode slightly above chance. So, as you might be able to deduct by reading this part of the presentation here, the null hypothesis should be generated by shuffling the labels of the groups and then applying the same classification procedure. So essentially, rather than having on every cross-validation fold the label for the image that they were shown before mental rotation and after mental rotation, you simply shuffle the labels of these two groups, repeat your decoding analysis. If you wish, even repeat the cross-validation procedure, and then you would generate a null distribution of decoding accuracies and compare the true data versus the null that you generated. It's really important to do this because it's just impossible to tell with a lot of these procedures what the chance level truly is, especially with the data sets that you're collecting, which may have some inherent bias in them. Additionally, even though we talked about the fact that shuffling only gives you inference from your own sample, that is already true of the decoders that we're doing. So this is really just increasing the power of the statement that you're making rather than decreasing it, because from what I see here, I think I would not be able to generalize these results to a population, not just because The statistics might have been done, a little bit with a little bit too many assumptions, but also because there's enough noise in these types of data sets that this small effect might not replicate. And as we talked before with the hypothesis testing lecture, it's important not just to report all of these results of these tests, but also to report your effect size and take that into consideration too. Anyway, I'm kind of just going on a rant, but I just wanted to wrap up this whole statistics section with a more holistic view of how you should think about all of these methods together. Okay, so as a little summary to this section, here's some things that you should remember about randomization tests. First, you need to really think carefully about the comparisons that you're making and what the test statistic represents before you make a null distribution for it. For example, with the first graph that I showed you with the unequal temporal bins, you could shuffle your data in many different ways at many different levels, and that's often true. So you need to think carefully about where you want to shuffle and what that shuffling produces, and whether that null distribution that you produce will be a sensible way to compare your data. Secondly, you then have to shuffle the labels between the key comparisons that you're making. So again, like I said, there's many different levels at which you can do the shuffling, but you always need to make sure that the shuffling at least occurs at some level which will affect the key comparisons that you want. So here's an example that you might run into one day. If you're calculating a randomized p-value for a one-way ANOVA with subjects as a random factor. So just to clarify, what this would be is, let's say time is the factor in your ANOVA and you have 10 different subjects, and you just want to say, there might be random variance in this data just because each subject is slightly different. So subjects becomes a random factor that's supposed to be a factor of non-interest, and time becomes your other factor. When you shuffle for this data set, you have to shuffle within each subject to maintain subjects as an intact factor. So let's say you have 50 trials for each of the 10 subjects. You would shuffle the 50 trials and shuffle the labels for those things for subject 1, and then do the same procedure for subject 2, and use different shuffling indices for every single subject. Otherwise, you wouldn't be able to maintain subjects as a random factor in your analysis. So, that comes to my third point. You really need to include every single detail about your shuffling procedure when you do a randomization or permutation test, because it really is important to the final interpretation of the data that you generate. And as with all of these hypothesis tests that we've talked about, think very carefully about the meaning and the interpretation of the p-value that you report. And like we saw in the example with the classification study, just because the p-value is small in your data set doesn't mean it will be small in another data set. One, because you cannot generalize very easily from the results of a randomization test. And two, because depending on the procedure that you choose, for example, to do something like classification or to calculate some measure like proportion, it really can depend a lot on the experimental setup that you have or the techniques that you choose that someone else might try to do somewhat slightly differently when they run a different study on the same concept. So all of these things are important when you write your discussion, when you write results, and important when you think about your data analysis in general. Okay, so just as a summary for this part, randomization tests are very flexible and super customizable, but they can be quite complicated. And I've just touched on a few of those topics today, and there's quite a bit more if you want to learn more. All right. So the last part that I wanted to talk about was model validation. And this is just a necessary topic if you're ever talking about resampling methods. I talked about all those Monte Carlo methods at the end of the last lecture, and we didn't go over a ton of them. So this is how you ensure that your model really reflects your data. This could be a statistical model that you generate, a model that you fit, like a general linear model, or it could be any type of model. So for example, just fitting a receptive field to a cell, for example. And you might ask, are there statistics for this kind of stuff? Sort of. You have to think of it in a slightly different way. And so we're going to talk about some of those things in the next few minutes. So essentially, the first question that most people want to ask is, how well does my model describe my data? And what they really should be thinking is, how well does a model predict my data? So describing and predicting are slightly different concepts. So you might have remembered from Vlad's lectures that you get an R-squared value when you fit something like a linear model. And the R-squared value describes a proportion of the variance in the data accounted for by the model. And it is a descriptive measurement of this fact. But you really want to know whether the model generalizes to other data sets. Like we were talking about with the p-values, that's really a central part of some of these questions that you're asking. And the first thing to start with is to figure out whether your model is generalizable even within your data set. So can it even predict your own data? And we'll talk about how to do that in just a second. And the other question that you need to address is this model parsimonious? Does it overcomplicate the true reality of whatever measurement that you're getting? Or are you just really boiling it down to the very essential bits? Okay, so first, is the model generalizable? So this is a pretty intuitive concept, but really important to stress because people do still get it wrong. So you want to train your model first on some data that is independent from the data that you tested on. So when you try to generalize within your own data set, you got to train it on part of it that is not part of the test set. And this whole concept is called cross-validation. And there's tons of different ways to cross-validate your model, but it always hinges on separating the training data from the testing data. So if you train your model on the same data that you tested on, you're essentially just, you're really just overfitting. So it would be like if you calculated a p-value for the null hypothesis that your samples are the same, and you just duplicated your two samples and then said, are they the same? Well, you would see that all of the measurements, like the mean and the variance, were the same between the two samples that you have because you literally just doubled it. That's the same exact thing that is true of models. You need to train the models on separate data sets. So one example of a cross-validation technique is something we actually already talked about, jackknifing. This is often called leave one out cross-validation. So in the same way, in the same procedure that we discussed before, you would simply take one trial or sample out of your entire data set, train the model on the remaining trials, and then test the model on the held out sample. And you would repeat this as many times as you have samples. So this is called a full cross-validation. And a more general method is just referred to as k-fold cross-validation, or leave k out. So rather than leaving one trial out, you could leave 20 trials out, you could leave 50 trials out. It really depends on how much data your model might need in order to accurately formulate some sort of guess. So for example, in some of the data that I have, I couldn't actually do a leave one out cross-validation technique because I need at least 20 to 50 trials in order to estimate my model. So my test data would need to be at least that big. And then finally, there's a more general technique that's called Monte Carlo or resampled cross-validation. So rather than doing an exhaustive cross-validation where you leave a subsample out and exhaustively rotate through all of the subsamples, you simply resample randomly throughout your data set. So you can resample half of it or 2/3 of it, or however much you want. And then simply test your data on the remaining part of the data set. And you do this many, many times. So the more times that you do this, the more it converges towards k-fold cross-validation. But a nice thing about this technique is that if you really do require a lot of data to create your model, you don't need to do an exhaustive cross-validation, which might take a long time if you have a ton of trials or a ton of samples. Instead, you could do, let's say, 1,000 Monte Carlo iterations and get pretty similar and similarly precise results. So let's go through a little example. Here I'm doing a very simple linear model. I have some data on the x-axis and some data on the y-axis, and I'm simply trying to predict the y data given a linear model of the x data. So I have this multiplicative factor m times x plus b, and I want to find m and b to describe my model. So if I actually fit a model to this and derive a value for m and b, I get a curve that kind of looks like this. And you can see that it's curved upward and it's really influenced by these 2 points out here, which are really look strongly correlated. But if you were to cover them up, you would see maybe in this little section here that there's not a huge strong relationship between these two values. So these guys are probably driving up our r-squared fits, which is currently at 0.33. It's not huge, but for the model output, it actually spit out a pretty low p-value here. So a reasonable thing to do here would be to try some sort of cross-validation technique. So I just chose Monte Carlo cross-validation. If you did jackknifing or some sort of leave k out cross-validation, you would get pretty similar results depending on the size of the data set that you pick. So I just split the sample randomly in half, fit the linear model to half of it, calculate r-squared, and then I applied that model to the other half and then calculated r-squared. And then I repeated this 500 times. So when I did this, I got some r-squared values which were in the range that I originally got, which was 0.33, you'll remember, and r-squared typically ranges from 0 to 1. So you'll see there's, a good amount, maybe 300 of the 500, which are here. But the remaining iterations are actually producing negative r-squared values. And you might say, how does that even make sense? It's r-squared. How can you get a negative with a squared value? You actually can, because in this case, an r-squared is not just an r value squared. So what a negative r-squared value means is the model was actually worse than a flat line in most cases. It explained less variance than the variance in the data set itself. So here's a little example. Here's this data that clearly has a positive monotonic relationship, maybe looks kind of like a square root function or something, and the fit, for whatever reason, is this straight line that goes in the opposite direction. In this case, if you were to calculate r-squared, it would also be negative. So r-squared is typically negative when the model actually creates more variance in the data than it explains. So it just generates a bunch of predictions which are completely off and don't really account for any variance in the data. So, oh, one of my pictures went away. So yes, the results of this cross-validation are showing me that although sometimes the model describes the data okay, and the other times the model is completely wrong. So this would lead me to suspect that fitting a linear model in this way isn't quite the best way to describe the data. So just as a summary here, to test a model's generalizability, you want to cross-validate. So that brings us to the second question. Is your model parsimonious? If you overfit your data without cross-validating it at all, you can always get an R-squared of 1. You would just have a predictor for every single data point. It wouldn't be too hard to do, and you could report that you have a crazy high R-squared and associated very low p-value. But as you know, this isn't quite what you want to do because it doesn't help anybody to use a model that is completely overspecified and is only true for your data set. So even if you cross-validate within your own data set, you can still get an artificially inflated R-squared because some predictors that you generate might not be meaningful. So if you have too many predictors, let's say one for every 2 data points, you still would have some interpretation problem there. So this isn't really quite a parsimonious model because there's probably a way to construct a model which has even fewer variables and which encompasses the effect that you have in a more theoretically sensible way. And in general, all people who fit models prefer more parsimonious models with fewer parameters. It's just much easier to interpret. So how do you do this though? You might think, I'm just going to fit a linear model, looks vaguely linear, but how do I pick the right number of predictors? Or maybe it looks nonlinear and you're not quite sure what shape of polynomial or function to fit because depending on the data you get, it looks different from subject to subject, for example. Luckily, there are some model selection criteria that you can apply when you don't know what else to do. And these are pretty good guidelines to follow. So one of them is the likelihood ratio test, which I'll go over all of these in detail in just a second. There's another thing called the, can't pronounce this, AIC information criterion, which people always just refer to as AIC. And the associated one, the Bayesian information criterion, which is called BIC. All of these tests basically penalize the model if it has a large number of parameters. And they all do it in slightly different ways. AIC and BIC are pretty similar to each other. But first I'll talk about the likelihood ratio test because you actually might have seen this before. So likelihood ratios can only be used for nested models in which you add new predictors on top of an existing model. So for example, you might have a model that looks something like this, and a nested model would have many of the same predictors and at least one different predictor. So here, the A1x1 term is the same, and so is the B term. But we have this additional term, A2x2. So the first model is nested within the second one. And then further, the second one is nested into this third one. And it keeps going. You can even have nonlinear relationships. For example, square this x1 factor in the next part. And the likelihood ratio is just a comparison of the likelihood for your null model over the likelihood for your alternative model. And you basically will take the natural log of both of these things and then calculate a test statistic D for it. And likelihood here is just really a term to describe the probability that this model is true. So you can continue to calculate this for all of your nested models. And then your D is compared against a huge family of, again, probability distributions for that test statistic. Excuse me. And you can just look those up. Finally, nested models are also pretty good candidates for ANOVAs. If you have a nested model, you can usually parse out the data so that the new variable that is added is an additional factor. So you can see whether the addition of that new variable accounts for any more variance. And as you know, the ANOVA does all of this simultaneously. That's not always true for all nested models. Sometimes you'll have to turn to the likelihood ratio. However, there are tons of models which are not nested. And the thing that people will really turn to here is the information criteria. So here I've just put up the equations. It doesn't really quite matter what they are. The results of the AIC and BIC criterion are pretty similar to one another once you do the entire process. So importantly here, they both share these two parameters, K, which is the total number of parameters in your model. So for that GLM we ran earlier, that would be M and B, those two parameters that we fit. And L is some type of measurement of the likelihood of the model. So just as I explained before, the model likelihood is simply just the probability that that model is true or fits the data well. And AIC and BIC use L in slightly different ways. You'll notice this one has an L hat versus this L here. And you calculate these criteria for all of your different types of models with your different numbers of parameters, and you'll basically want to use the model with the smallest AIC or BIC value. So the more parameters you have, the larger K is and the larger the value gets. And this is modulated by this likelihood factor. So if it's super likely, but you have a high number of parameters, you'll still get a somewhat small BIC or AIC. So this is pretty cool. You can look up more, and I can send you some references if you're interested in using this in your own data set. Okay, so how do you pick the right number of predictors? There's all these model selection criteria that I just reviewed, and luckily, you actually can just use simple built-in methods in MATLAB to get a lot of these values. For example, with the linear method fitting methods that Vlad reviewed in the previous lectures, fit-LM and fit-GLM, the outputs actually have an ability to spit out the log likelihood and the AIC and BIC, which is pretty cool. So the new statistics package in MATLAB is pretty awesome. All right, so to find a parsimonious model, calculate some penalties for the number of parameters. All right, finally, I'm just going to do a brief aside on curve fitting, and this is just bonus content for someone who really wants to learn about curve fitting, because we realize that we haven't quite covered it in the material for this class, and it can be quite important to some disciplines. So for those of you who want to fit anything that's not a linear model, there's a great huge suite of tools to do so in every data analysis language that any of the comp neuro people prefer. So that could be MATLAB, Python, R, any of those things easily can do curve fitting. In particular in MATLAB, there's a bunch of curve fitting tools such as cftool and a bunch of different algorithms for searching all of the huge parameter spaces that you have to find the model with the smallest error. And at the very end of this video, I'll do a little demo of cftool. It's pretty intuitive to use, and you can just enter the form of the function that you desire to fit in your data, and then it'll spit it out for you. It's pretty cool. So, briefly, in order to fit a model, you basically try out a bunch of different parameters and you calculate the error between the model that you have and the data that you actually measured. So if you fit a line here that's just right through the middle of this data cloud, the error that you would get would be pretty small because all the points would be really close to the line. If you drew the line the other way, that would not be true, so you would get a large error. So in general, all of these fitting algorithms tend to want to minimize the error when they're doing their search. And I'm using some graphics from a pretty neat post that I found on what looks like a blog, Atomic Object. So I have the reference at the end if you'd like to read the whole article. So as a very high-level conceptual overview, the way that a lot of these algorithms perform the search is through gradient descent. So basically, this is the 2D, well, the 2D sheet that is then folded into a 3D error surface for the data that I showed you previously. So the two dimensions here are x and y. And here I'm showing you that the things that they're trying to fit, the slope and the intercept, are on the bottom axes here. So this is the entire parameter space that is being searched here. And for example, for a slope of 2 and an intercept of 0, the error would be this value that you see here. And the error is both encoded on the z-axis and color-coded on this sheet. So the brighter red is a higher value of error, whereas the blue is a lower error value. So basically, imagine dropping a marble at the top of this surface and just watching it roll down this surface. It's essentially what you want to simulate when you're doing a gradient descent. Marvin will talk about this a little bit during his lecture because gradient descent involves calculus, and you would need to define the gradient along your error surface. So essentially, all of these cool little algorithms define a gradient for the data you have, hopefully, that's the easy way to do it, and then tries to start out at random points in the sheet and drop the marble down to find the lowest spot. And you can imagine if you don't have a cute little easily predicted data set that looks like this, your surface would probably not be so smooth or easy to find the minimum of. And often for a lot of data sets, there are many local minima where your marble might get stuck and wouldn't be able to get out to find the really true lowest point. And then you would just try all different points of dropping your model or your marble, which would be all different points of starting out your curve fit. Anyway, that's just a brief little aside. If you want to know more about gradient descent, this blog post is pretty interesting, and I can direct you towards a lot more resources for that, including a different lecture. Okay, so now we'll go to our brief cftool demo. Okay, so we're in MATLAB now. You can see in my workspace to the left that I have this variable x and y, and that's going to be the data that we want to fit. So in the command window, all you want to do is type the words, or the letters, CF and then tool, which stands for curve fitting tool. So this is just the graphical interface, or the GUI, that allows you to interact with a bunch of different fitting functions in MATLAB. It's super easy to use. You can find out all the things behind it that it's packaged with after you get a little experience with the cftool. cftool is great because it's got this little window that pops up, so I'm just going to maximize it here. It already knows what's in your workspace. So under X data, I'm just going to select X, and under Y data, I will select Y. So now you can see that it's plotted the data here for me. So on the x-axis is x, on the y-axis is y, and for now it's just fit this single line because it's auto set to polynomial with a degree of 1, which is just a line. And you can see here that the data actually don't quite look like that. It's got a little bit of a curve to it that peaks around 4 to 6 for x. So polynomial of degree 1 is probably not the right thing that you want. So you can see here, if you scroll down, there's all kinds of different equations that you might want, like a Gaussian, an exponential, any type of polynomial, a power function, a spline, an interpolant, or you could even have a custom equation. So here you can just type in whatever function you want. So let's see here, I have this exponential function, and I'm fitting the constants a, b, and c. And you'll see every time it fits, on the left here, it spits out a bunch of data, including the r squared value, the sum of squared errors, and even the root mean square error. And even shows you the 95% confidence intervals for each of the coefficients that are fit. So, you know, a here is a factor of around 1,983, but it varies between these two values. So you can see here at the bottom, it even gives you a warning about how a negative r squared is possible, which we just talked about. So here I'm going to type in just a simple little function. All right, and so I've put in a little function that's just like a squared, you would see it as a parabola if it was zoomed out even more. So here it's tried to fit this little parabola. It hasn't done a great job here. Let's see if I can get this a little better even. So now I'm telling it to fit any type of power function that it wants, and it's even given me a slightly different line. It's actually wanting to go flat because all this stuff at the bottom is close to 0. So let's go to our polynomial function and give it 3 degrees. So now I can see it does a pretty good job. It fits this curvy little function here, and the r squared is pretty high. It's 0.85, and that seems to be a good approximation for our model. And so as a little reveal here, this is just some data from the Poisson-Rand function, and it should look roughly Poisson. And it kind of does. There's a little bit of randomness because I didn't sample a huge number of points. I think I only have 15 or something like that. So a third degree polynomial is perfectly fine to describe this function. Okay, cool. You should play around with cftool on your own time. It's pretty neat. And yeah, so we'll go to the end of the video. All right, thanks for listening to this little bonus lecture. Here are the references for a couple of things I talked about, including that Nature paper with the time point data, as well as that blog post explaining a little bit about gradient descent and linear regression. Thanks for listening. Bye.